Welcome students. This is our third topic of the unit. In this topic we are going to see parts of cell and their function. The objective of the lesson is at the end of this section you should be able to name the different parts of cell and explain their function. Students, after you have learned or after you have studied this topic, you have to name the different parts of cell and explain their function. The second one is compare the Davson-Danieli and fluid mosaic model. At the end of this section, you should be able to compare these two membrane models. And the third one explains the mechanism of substance transport across a cell membrane. So these three are important points that you have to remember. When we start the lesson, it starts from parts of cell and their function. When we talk about cells and their function, you have to imagine cell as a structure with a cell membrane that separates the cell content from its environment, and next to the cell membrane there is a structure known as cytoplasm, which is a fluid matrix that contain different organelles and different dissolved substances. And also you have to remember that when we talk about cell structure, there are few organelles or different organelles in the cytoplasm. Therefore, when we study parts of cell and their function, we have to know what organelles are. Organelles are cell structures with a specific function, and these organelles are found in the cytoplasm of the cell. They are found in the cytoplasm of the cell. And this cytoplasm, as I have told you, contain organelles and some other dissolved substances. When we talk about the structure of cell, we only consider the structures, but here we have the diagram of animal cell that you can observe how the structural arrangement looks like in the animal cells. And as you see here, in animal cell, the nucleus is found at the center, relatively central part of the cell, and the nucleus has different components. And in addition to the nucleus, here we have the endoplasmic reticulum, that is an extension of nuclear membrane, and it's also found in the cytoplasm. Next to the endoplasmic reticulum, we have Golgi Complex or Golgi apparatus, and we have mitochondria, which is one of the important structure. And you can have different other structures with their specific function. But when we come to plant cells, what makes difference? In plant cell, the nucleus is found near to the side. This is because the central part is already occupied by large vacuole. Here it is vacuole, and this vacuole occupies the central part. For that reason, the nucleus is found near to the side of the cell, but the other structures have almost similar function. Let's start from nucleus. Let's start our study from nucleus. The nucleus of a cell is considered as controlling center of the cell. It is controlling center of the cell, and if you look the structure of nucleus, this one is the structure of nucleus, and this nucleus has three components. It has three components. The first one is nuclear envelope or nuclear membrane. This one is the nuclear membrane, and the nuclear membrane, as you see, is not smooth. It is interrupted, or it becomes discontinuous due to the presence of small pores known as nuclear pores. the nuclear pores are important for exchange of material between the nucleus and the cytoplasm. Next to the nuclear membrane, we have thread-like structures. These are genetic material or the chromosome that stores all information about the cell. And here we have a dense circular or spherical structure that is nucleus, which is found in the nucleus and responsible for the synthesis of ribosome. Here we have the explanation. The nuclear membrane is a double membrane. It's a double membrane with pores. As I have said, it has pores on its surface, and these pores are important for exchange of material. The nucleus are site of ribosome synthesis, and the chromosome contain genetic material of the cell. And the next structure is mitochondria. Students, mitochondria is known as the powerhouse of the cell. Its other name is the powerhouse of the cell. And if you look at the structure of mitochondria, mitochondria has outer membrane. This one is the outer membrane. And as you see, the outer membrane is smooth. And the second one, the one that is folded, is the inner membrane, which is folded. And inside the inner membrane, there is a liquid matrix that is site for set of chemical reaction. And the name of this folding in the inner membrane of mitochondria is known as cristae. This folding is very important because it increases the surface area for chemical reactions that takes place on this membrane. So the powerhouse of the cell or the mitochondria has such a structure, which is also membrane bounded, and the membranes are double, and also the inner part of the mitochondria consists of matrix that is site for different chemical reaction. The matrix, in addition to this, has circular mitochondrial DNA. It has DNA, RNA, protein, enzymes, and lipids. We will talk this when we study cellular respiration. So the function of mitochondria is it is site of energy production. Cells will obtain energy from their mitochondria. The mitochondria is site of energy releasing. The next structure is chloroplast. Chloroplast, its other name is kitchen of the cell. Can you guess why we call chloroplast kitchen of the cell? Okay, it is directly related with its function. Let's see the structure of chloroplast. The chloroplast, as you know, is one type of plastid. You have learned about plastids in your lower grade. These are structures only found in plant and algae. And plastids are 3 in type, and here we have one of plastid known as chloroplast. Chloroplast contain green pigment. It contains green pigment known as chlorophyll, and chloroplast, due to the presence of chlorophyll, it plays important role in photosynthesis. Structurally, chloroplast has 2 membranes, outer membrane and the inner membrane. Inside the inner membrane, there are thylakoids known as thylakoid membranes. These thylakoids are site of light reaction, or the first step of photosynthesis occurs in the thylakoid. And the other one is the inner membrane contain liquid, semi-liquid part. That liquid part is known as stroma. It is known as stroma. The matrix is called the stroma, and stroma has membranous sacs called thylakoids. The thylakoids, as I have told you, they are site of photosynthesis. Mainly the first stage of photosynthesis occurs in the thylakoid. So what is the function of chloroplast? It is site for photosynthetic reaction. That's why we call chloroplast the kitchen of the cell, because photosynthesis is one way of producing food by cells. The next structure that we are going to see is Golgi Complex. Golgi Complex, the name is derived from the founder of the organelle, Camillo Golgi. For that reason, it is known as Golgi Complex or Golgi apparatus. Golgi Complex is also considered as the cell post office. You know why? When you want to send a different message or different package by using post office, the first point is to pack the material or the message. After that, you will make it a tag on its on the envelope. After that, it will be transported. That is what you observe in case of Golgi Complex. Golgi Complex, structurally, it is a single membrane organelle, and the single membrane is folded. As you see here, it is folded and form cisternae. The folding in Golgi Complex are called cisternae, and this organelle is found both in plant cell and in animal cells. The Golgi Complex, when we look the function of Golgi Complex, it receives, pack and ship or transport vesicles coming from the endoplasmic reticulum. Or in other words, the Golgi Complex is important to pack, to transport, to modify different cell products that are produced by endoplasmic reticulum or other part of the cell and return back to endoplasmic reticulum. In addition to this, the Golgi Complex is site for lysosome production, because in Golgi Complex, as you see here, after the material is processed in all these steps, finally it becomes released in the form of vesicle. One type of vesicle that can be formed in this form can be lysosome. So that we can say that Golgi Complex produce lysosomes. And it secretes cell wall. In case of plant cell, cell wall is secreted in Golgi Complex. The next membranous structure that we are going to see is Endoplasmic Reticulum. Endoplasmic Reticulum, as you see on the diagram, they are extensions of nuclear membrane, the nuclear membrane extension, and structurally they may have different additional components. They may have ribosome, or some of them may lack ribosome. For that reason, we study endoplasmic reticulum by grouping into two as a smooth endoplasmic reticulum and rough endoplasmic reticulum. The smooth endoplasmic reticulum is type of endoplasmic reticulum that lacks ribosome. That's why it is known as a smooth. And its function is synthesis of lipid, metabolism of carbohydrate, and detoxification of drug and poisons. These are the functions of a smooth endoplasmic reticulum. And the next one is rough endoplasmic reticulum. Rough, the name rough is given for this structure is due to the presence of ribosomes. Ribosomes are found on the surface of rough endoplasmic reticulum, and their function is protein synthesis and transport. They are involved in protein synthesis and transport. Therefore, the endoplasmic reticulum is one type of membranous structure that involve mainly in producing different products and in transporting cellular products to different part of the cell. The next structure is ribosomes. Ribosomes are known as protein factories of the cell. From this, you can guess the function of ribosome. Ribosomes, if you look in every type of cell, they can be found in different part of the cell, but generally ribosomes can be found in the cytoplasm. Simply, they can be found in the cytoplasm, or they can be found in inside the mitochondria, or in chloroplast or attached on rough endoplasmic reticulum, as I have said earlier, and they are also attached on nuclear membrane. They are attached on nuclear membrane. Structurally, ribosomes have two protein subunits, a small subunit and a large subunit, and their main function, as its name indicates, they are important for protein synthesis. And when protein synthesis takes place, the large subunit and the small subunit come together and read the messenger RNA and lead to the formation of proteins. Therefore, their main function is site of protein synthesis. They are site of protein synthesis. That's why every cell has ribosome, because unless they produce protein, cells cannot perform any metabolic process. That's why they are found in every type of cell. The next structure is vacuoles. Vacuoles are vesicles. They are vesicles, and they are also single membrane bounded vesicles. Structurally, these organelles are found in plant cell. They are found in plant cell, but they are absent in animal cell. If you look at plant vacuoles, they are large in size, here as you see, and they contain most of the central part of the cell, and this central part or central vacuole contain different dissolved substances in it in the form of solution, and we call this dissolved substance or solution cell sap. The cell sap is contained in a membrane known as tonoplast. Vacuolar membrane is known as tonoplast, and the cell sap, as I have told you, contain dissolved salts, sugar, enzyme, and so on. These are found in vacuole. The next structure is lysosomes. They are known as suicidal bugs. This is because of their activity in cell. Lysosomes are single membrane bound vesicles. They are single membrane bound vesicles filled with hydrolytic enzyme. Can you tell me what do you mean by hydrolytic enzyme? Okay, hydrolytic enzymes are digestive enzymes that are found in lysosomes, and lysosomes are found only in animal cells. They are characteristics of animal cells. They are produced from Golgi complex. They are produced from Golgi complex. As we have said earlier, the Golgi complex can perform this action. And when we come to the function of lysosomes, they are used in intracellular digestion, which means digestion that takes place inside the cell, and they are used for destroying old and non-functional cells. Old and non-functional cells. They recycle worn out cells. They recycle worn out cells. Here we have the diagram that slightly indicate the action of lysosomes. In this diagram, it shows phagocytosis, or the action of lysosomes when the cell takes large food particles and digests it. The first point is the food particle is engulfed by the cell, and then food vacuole will be formed. This food vacuole will fuse with lysosome, as you see here on the diagram, and the lysosome release the digestive enzyme to dissolve or to digest the food particles. After that, the cell will use the end products. And on the other side, it shows a process of autophagy, which means when lysosomes break or digest cell organelles. When the lysosome digests cell organelles, here we have old mitochondria that is contained in vesicle, and then lysosomes will fuse with the vesicle and release the digestive enzyme that can break down the mitochondria into different pieces, so that the cells can use lysosomes to avoid old organelles. This has importance because if the organelle is old, its efficiency becomes low. For that reason, it must be removed from the cell so it can leave a space for newly formed organelles. Secondly, if cells get food particles in this mechanism by phagocytosis, the food particle must be digested and make ready for use by the cell. All these actions are done by the action of lysosome. The next structure is cell wall. Cell wall is the outermost layer, non-living and rigid. It is the outermost layer and non-living and rigid, and this structure is found in bacteria, in bacterial cell. It is also found in fungal cells, or it can found in it also found in plant cells. If you look at fungal cells, the bacterial cells, bacterial cell wall is made up of peptidoglycan. This is your lesson on Unit 2. Bacterial cell wall is made up of peptidoglycan, whereas fungal cell wall is made up of chitin, and plant cell wall is made up of cellulose. That's why we call plant cell wall as cellulose cell wall. Well, in all cases, the cell wall is permeable. When we say permeable, it is a structure that can allow movement of substance in both directions. Freely, substances can move in and out through the cell wall. And here the structure shows plant cell wall, which means cellulose cell wall. The cellulose cell wall has such type of crisscrossed arrangement of cellulose molecule. This arrangement give plant cell wall to have elastic nature. It can able to stretch when the cell gets more fluids or more water into the cytoplasm. That's one of the importance of cell wall in plant cell. Here it says the cellulose, plant cell wall is made up of cellulose. In bacteria, it is made up of peptidoglycan. In fungus, it is made up of chitin. And its main function is rigidity. It gives rigidity for plant cell, for example, because when plant cells get turgid, when they get more water into the cytoplasm, they become swell, and the swelling in plant cell give rigidity for the plant due to cell wall. Why? As I have said, cell wall is elastic in nature, mainly cellulose cell wall is elastic in nature. Due to this elastic nature, it cannot be break due to pressure from inside. Mechanical support and protection for plant cell. It gives mechanical support and protection for plant cells. These are the function of cell wall. The next structure that we are going to see is the cell membrane or plasma membrane. The cell membrane present in all cells. It's present in all cells, and when we look at plant cells, the cell membrane is found below the cell wall, and when you look at animal cells, it is the outermost layer or the outermost membrane in case of animal cell. Why? Because animal cells have no cell wall, therefore the cell membrane is considered as the outermost membrane. In all cases, whether they are plant cells or animal cells, cell membrane is semi-permeable. Semi-permeable means it can allow some particles to cross into the cytoplasm or out of the cytoplasm. Or in other words, when we say semi-permeable, it can pass particles selectively to cross it. It can allow particles selectively to cross it. That's why most of the time when we talk about the action or the function of cell membrane in cell, water can move in both directions freely, but other important molecules like protein cannot easily leave the cell or enter into the cell. This is because the cell membrane is a semi-permeable, or it is selectively permeable membrane. Selectively permeable membrane that select materials crosses the membrane. If you look at the structure of cell membrane, it is made up of phospholipids. From your lesson on Unit 2, Biological Molecules, you have learned that phospholipids are organic compounds that are basically make all biological membranes. Therefore, cell membrane, as you see, it is made of phospholipids. These are phospholipids. And the other one is protein. The plasma membrane has protein. For example, if you take this, these are protein molecules that cross the plasma membrane, and they have different function. Carbohydrates, as you see here, these are carbohydrate molecules that make the plasma membrane and have different function, specific function. And lastly, the cell membrane has cholesterol. Cholesterol is important in maintaining the fluidity of the membrane. So it is found in cell membrane. Okay, up to now we have seen the structures that are found both in plant cell and in animal cells, and now we are going to summarize the main points of the lesson. The first part that we have seen in this period is different parts of cell. We start discussion from the nucleus, which is the central part for animal cell that control all activities of the cell. When we say this, nucleus, whether the cell is plant cell or animal cell or fungal cell or algal cell, its main function is to control all activities of the cell. You know the reason? In the nucleus, as we said, there is DNA, or the genetic material. This genetic material store all information, basically metabolic processes that occurs in the cell are controlled by the instruction of DNA. That's why the nucleus is controlling center of the cell. After that, we have seen mitochondria, the powerhouse of the cell. This is because mitochondria is the site for cellular respiration. Cellular respiration means set of chemical reactions that release energy from the food that we take. And this reaction occurs in the mitochondria, and it is known as the powerhouse of the cell. And the other one is chloroplast. As I have said, it is site of photosynthesis, and the inner part that contain the thylakoid is the liquid stroma. In case of mitochondria, the inner part is matrix, and the matrix is site of cellular respiration, especially aerobic respiration. When we come to chloroplast, the thylakoids are site of the first step of photosynthesis, and the second step of photosynthesis occurs in the stroma, the liquid portion of chloroplast. So you have to list difference between these two structures, even though they have some similarity, but you have to list the main difference between chloroplast and mitochondria. And the other similarity, both have their own DNA. They have their own DNA. That means they are able to produce their own protein. This is because of mitochondria and chloroplast. They were prokaryotic cell in ancient time before evolution of eukaryotic cell. For that reason, they have their own DNA. After we discussed the mitochondria and chloroplast, we have seen different membranous structures like Golgi Complex, Endoplasmic Reticulum, vacuole, Lysosomes. All these are membranous structure in cell with a specific function. So we have to list their function in more detail. If you took, for example, Golgi Complex, it is used for packaging, transporting, or modifying cellular products. That's why it is known as the post office of the cell. The lysosomes are suicidal bag. That means suicidal action takes place due to the action of hydrolytic enzyme in lysosome. But the hydrolytic enzymes can undergo different reaction in cell for the cell advantage. As I have told you, old organelles must be removed so that new organelle will be replaced. New products or different food particles can be taken by the cell, and this can be processed by the cell for different activities. And the other parts that we have seen are the ribosomes, which are site of protein synthesis. And I have told you that ribosomes are found in every type of cell, And lastly, we have seen cell wall, which is found in plant cell, fungal cell, and in bacterial cell. But in all these three types of cells, the cell wall is made of different components. So you have to remember this component. And the other structure is the cell membrane, which is also found in every cell because it contains all the cytoplasmic content, and it separates the cell from its environment. So you have to see the structure of cell membrane that makes it selectively permeable because of the presence of these different components. Students, this is all for this period. Next time we will see the function of cell membrane in more detail. Thank you.